Ever stop to marvel at how your brain, you know, effortlessly picks up new skills or remembers faces without just wiping the slate clean?
It's pretty incredible, isn't it?
Yeah. So today, we're plunging right into the heart of that, how our brains actually form and, crucially, connect memories.
Uh-huh. We'll be looking at some really interesting new science.
Exactly. Stuff that's actually tracked brain cells during learning based on work from researchers like Dr. Thomas Ryan and Dr. Clara Ortega de San Luis.
Right.
And our mission really is to get a handle on the mechanisms that make our memory so flexible, but also so stable.
And it's fascinating because, for a long time, the debate was, is memory stored in single cells, or is it about the links between them?
That old question.
Yeah, and this recent research really shifts the needle quite strongly toward the connections being key.
Okay, let's jump into that then. This idea of enneagram cells, what are they exactly?
Well, Dr. Ortega de San Luis describes them as groups of brain cells.
Mm.
And when you have a specific experience, these cells get activated, and they actually change themselves somehow to incorporate and, hold that information.
So it's not like one single neuron is a little recording device for one memory, like the old way of thinking.
Not really, no. That was sort of the earlier idea, like tiny storage units.
Mm-hmm.
But the picture emerging now is much more networked.
A network.
Yeah, memory seems spread across communities of these enneagram cells. And the really important part, it's the connections between them. Those relationships are constantly changing. That seems to be the key.
That feels more intuitive somehow. And they actually watched this happen, right? With two different memories?
They did. They tracked two separate sets of these enneagram cells, each linked to a different experience.
And what happened?
Well, this is where it gets cool. When they reactivated one set, let's say the memory for event A,
Mm-hmm.
sometimes the other set, the one for event B, would also light up, show some activity.
Whoa. Like finding a related photo when you just meant to look at one specific album on your phone?
That's a great way to put it. It strongly suggests these memories aren't isolated islands. They're linked.
Okay.
Pathways are being built between these different groups of cells. Which brings us to the next big point.
Which is?
That memory isn't just about the cells firing, but about the connections they build. And, crucially, reshape over time.
These structural changes. They're needed so we can learn new stuff without just forgetting everything else.
Exactly. It prevents overload, prevents just erasing the old stuff. It allows for integration.
So how do you actually see the brain forming these connections? It sounds so abstract. You mentioned neuron labeling.
Yeah, it's pretty clever stuff. Researchers use genetic tools to basically tag the neurons that are active during one specific event, like putting a little flag on them.
Okay, flags on the active cells.
Right. Then they watch those same flag neurons when, say, the subject experiences a second, maybe related event.
And they're looking for.
They're measuring how well those flag neurons make new links, new connections with other cells. Cells may be tied to older, different experiences.
Gotcha. And they didn't just watch, did they? They tested if these new links actually mattered for remembering.
Precisely. That's where optogenetics comes in. Amazing technique.
Right, the light thing.
Yeah, you use light to control specific neurons. You can turn them on, turn them off.
So they could switch off these newly connected cells.
Essentially, yes. They could manipulate the activity of these connections with light and then see, does messing with this connection disrupt the memory recall?
And did.
If recall stumbled when they interfered with those connections, well, that's strong evidence that those specific links were vital for that memory.
That's incredible. So connections are crucial, but what controls how strong or stable they are? This protein, PSD95, you mentioned it acts like a gatekeeper.
Yeah, PSD95. It's a protein found right at the synapse.
The synapse being the connection point between two nerve cells.
Exactly, that little communication gap. Think of PSD95 as helping to anchor the receptor molecules there. It sort of reinforces the connection.
Makes it stick.
Makes it stronger, more stable. Yeah. But it plays a big role in keeping those connections solid, the ones that hold the memory.
And the researchers messed with this protein too, right? They lowered its levels.
They did. In neurons that were known to be part of storing a specific memory, they reduced the amount of PSD95.
And what happened? Did the memory just vanish?
Well, that's the surprising part. Sometimes, even with less PSD95, even when they were trying to kind of weaken the memory's foundation, the memory lingered. It didn't always disappear.
Huh. So what does that mean if the memory sticks around even when the gatekeeper is less active?
It suggests a couple of things. One, while PSD95 is important for like the day-to-day flexibility of connections, what scientists call plasticity,
Right.
a really well-established memory might have some built-in resilience. It's not solely reliant on maximum levels of this one protein.
So that could explain why some memories are so persistent.
It might hint at that, yeah. Why some things are really hard to forget. It also suggests that if PSD95 levels are disrupted, maybe through disease or something, it could definitely impact normal learning.
Making it harder to form stable new memories. Or maybe harder to update or forget old ones.
Potentially, yes. It points to how delicate that balance is for normal learning and memory function.
This whole picture seems to point towards the brain not just adding new files, but weaving new info into the existing network.
Yeah.
Updating, not just replacing.
Exactly. Dr. Ryan put it really well. He said something like, we should search for information between cells.
Between the cells, emphasizing the connections again.
Precisely. It's about the relationships, the network structure.
If you think about everyday life, that makes so much sense, like learning a new way home.
Perfect example.
You don't forget your old route instantly. You just add the new turns, the new landmarks onto your existing mental map.
Your map evolves, integrates the new information. You strengthen some connections, maybe weaken others slightly. It's dynamic.
So, thinking bigger picture,
Hmm.
what are the potential applications here? Understanding this memory updating process.
One really exciting possibility, though it's still early days,
Sure.
is maybe developing therapies. Therapies that could target these proteins like PSD95, involved in shifting the connections,
To do what?
potentially, to help gently reframe harmful memories. Think about conditions like PTSD or strong phobias. Could we weaken the emotional impact by tweaking the underlying network?
Wow, that's potentially huge, but complex, obviously.
Very complex, lots more research needed for sure, but it opens a door.
And it's not just about trauma, is it? This constant learning and updating is just life. Remembering names, finding your way around, picking up a new hobby.
Absolutely, our daily routines depend on it. And understanding these mechanisms could eventually help with things like age-related memory issues or maybe even learning difficulty.
Finding ways to support that flexibility and stability.
Right. And another interesting finding was that some neurons aren't just single taskers. They can play different roles depending on the memory or the context.
They can change jobs.
Sort of, yeah.
Mm.
Their roles can shift as we learn and experience new things. It just underscores how incredibly adaptable the brain is.
Okay, so let's try and wrap up the key takeaways from this deep dive. It seems like memories aren't just files in specific cells.
No, much more likely they're stored in the connections between groups of these enneagram cells.
And these connections are always changing, being built, reshaped as we learn.
Constantly. And proteins like PSD95 are crucial players in managing all that change.
And the big idea is updating, integrating new info into the old framework, rather than just overwriting.
That really seems to be the core concept emerging. The brain as a master integrator.
So for you listening, here's something to chew on. How might knowing that memory is about building connections change how you tackle learning something new?
That's a great point.
If it's all about the links, what new connections could you maybe consciously try to build today, relating new stuff to what you already know?
Yeah, being more intentional about finding those links maybe. It could actually strengthen the learning process.
Definitely food for thought. This has been a really, really fascinating deep dive into how memory works.
It really has. The science is moving so fast.
If you enjoyed exploring these neural pathways with us, please make sure you follow the deep dive.
We'd love to have you back.
Yeah, like and subscribe so you don't miss our next explorations. And hey, tell us what you're curious about. Send us suggestions for future topics. What should we dive into next time?